Well, thank you so much for the invitation to be here. It's a real pleasure to give you a glimpse on the research that we are doing in the lab. And I would like to start by explaining you a little bit of the context of this talk. So when Stefan emailed me to invite me to give a talk, he asked if I could give an update on the topics that I was presenting a couple of years ago in a conference in France, which is where we met. And I'm using on purpose the first slide that some of you have seen in other forums with this kind of enigmatic title. And that's why I've called it the sequel. So let me tell you a little bit more about what this means. So in that moment, we were working on some interesting ideas that were not so well explored in animal evolution. And we were using state-of-the-art methods such as analysis of chromosome-level genomes and protein language models to tackle questions that otherwise would seem a little bit like fictional, but they were actual science. That's why we call it like this. So the first story was about hopeful monsters. And unfortunately, I don't have a lot of time today to go into both topics, into both stories, but I would like to give you a teaser on the update. Long story short, this story was about large-scale genomic rearrangements. Now, as you all know, there are all these initiatives aiming at sequencing chromosome-level genomes of tons of non-model species. So we do have a lot of extra context that before we did not have with draft genomes or just with single gene families, right? So this is a picture that represents the macrosynteny of all these genomes. Each line is one species, each white square is a chromosome, and each color thread means sets of orthologous genes that have been traveling together throughout evolution. And what this is showing is that different animal phyla have highly conserved macrosynteny. They have a very similar group of genes that are maintained together across phyla, but then we can have these massive genomic upheaval events, even at the within phylum level. Now we are seeing them even at the genus level, and where all this conservation is broken. And then new colorful threads are originated as a consequence of that. So this is something that's been published a while ago. Many other people have been seeing these things. So we do know now that punctuated evolution happens in animals, in many animal phyla, and many different taxonomic and systematic scales. And the story that I'm just gonna briefly mention is that what we realized is that there are no methods to actually infer these colorful threads here in this scenario, right? So what we developed in the lab is a pipeline to try to infer them, not only in the tips, but also in the nodes, and being able to reconstruct this, let's say, collinear homology between chromosomes in the face of large-scale genomic rearrangements. Is this tool called Linguine. So for those of you that are interested in this topic, feel free to check it out. And we also developed a simulator of genomic rearrangements and episodes of whole genome duplication, gene gain, and gene loss that we call Garlic. Again, I don't have a lot of time, but I thought that this could be of interest to the audience in this room, and I will be super happy to show you some slides on this during the poster session or in a different way. So if you're interested, just feel free to search for me, and I will tell you what all these cool tools are about. And the second story is actually about genomic dark matter, and this is the one that I really want to develop today, a little bit also in line with the philosophy of this conference, which is giving a state-of-the-art introduction to the topic and then going a little bit deep into the methods. So what is this about? This is about a story that was obsessing me for a while, still obsessing me. We started to work on this together with Ana Rojas, who is a collaborator at the Andalusian Center of Developmental Biology in Spain, and we started in 2021. So what happens when you work in non-model organisms is that many of the genes do not have a functional annotation. So just imagine you sequence your genome of interest, and there's an expansion of genes that may be related to something cool or specific that this animal is doing, but then you don't have a functional annotation. So it's kind of a dead end. Or the same when you do, I don't know, a series of experiments and you have population A that resists higher temperature than population B, and then you do differential gene expression and you end up with genes and you don't know what they do, right? So this is a problem that we were facing, and this was the initial, let's say, scenario in which we started to develop this line of research. So, of course, I don't need to convince this audience that understanding gene function is important, because for us, people working on evolutionary biology that are driven by biological questions, it's extremely important to go towards the phenotype, understand adaptation, and so on and so forth. So how do we categorize or measure gene function? We can use motifs, domains, etc. But one very popular way of doing it is gene ontology terms, GO terms. As all of you know, gene ontology terms are a unifying nomenclature that we use in the scientific community that has a code, so we understand each other, right? And we talk the same language and at the same level. This has been a little bit annoying, so let me stop the PDF and so it's not cut. There we go. There are three categories of gene ontology. We can have cellular component on one side, then we can have molecular function on the other, and on the third layer we have biological process. And this is the way we try to figure out what our genes of interest mean, and we can build hypotheses that we can later on test, and so on and so forth. So how do we assign GO terms? All methods until very recently were based on sequence homology, right? So we had our protein sequence, we were identifying other homologous in reference databases such as UniProt, for instance, or ECNOC, and we were finding our homologs through sequence similarity, and if one of these sequences in the reference database had a GO term, it was transferred as a, let's say, putative function to our sequence, right? But the problem is that this, again, left many sequences without function, which was extremely annoying. So to give you an idea of the magnitude of this, I'm going to show you numbers. So we evaluated this in 1,000 animal genomes from all phyla, so 24 million genes. When I say genomes, could be also from transcriptomes, let's say the proteome, the repertoire of coding genes. And this is what we found. So these are all the animal phyla, and all this is a huge fraction of genes that do not have GO terms. And this is what we call the dark proteome. So it was like approximately 50%, just for you to understand how bad the problem was. Of course, this is getting more and more worrying, because now we have all these initiatives again, like ERGA, the Darwin Tree of Life, the Earth Biogenome Project, Atlas-C, and so on and so forth, aiming at sequencing more and more genomes, right? So the knowledge is there, but we just cannot unlock it. So in this context, in 2021, we started to test protein language models. It was right before ChatGPT, when people were not so familiar with this, to try to tackle this question. And that was an idea indeed of Ana Rojas, my collaborator, who is a bioinformatician working in functional genomics. We started to work together, and she threw a few keywords, protein language model, embedding, that I had never heard in my life, And she told me, this is the future, we have to go for it. So just a couple of basic things on how language models work. So if we try to compare two sentences, Peter and Mary bought a house in 2020. Dimitris and Teresa rented a boat. This was supposed to be a ship last summer. Copy paste, you know. If we try to align them, there are no words that are similar to each other, right? However, if we take into account the grammar, we understand that Peter and Mary are the subject, they are names, they are equivalent to Dimitris and Teresa. We do have a verb, we have an object, and we have a date, right? So these are some local properties in this grammar, but there are also some global properties that we all understand because of context, such as the topic, the language, the sentiment, and so on and so forth. So researchers, mostly of them working on biophysics and chemistry, they started to use this in proteins as well. They thought, okay, this could be very cool as well, to try to encode the grammar of proteins without relying on the sequence, right? So you can think on a similar example with sequences. There are some local properties, such as phosphorylation points or active sites or disulfide bonds and so on and so forth. But there are some global properties as well, such as order. We were talking before about intrinsically disordered parts of the proteins, organisms, subcellular component, where they are, and so on and so forth. So the idea is to leverage exactly the same architectures and philosophy that we are using in language models in protein language models. And what these models do is to convert the sequence into a multidimensional vector that is called an embedding. So this is a high-dimensional vector that represents the sequence. Could be a protein, could be a chunk of the genome, depending on the model that you use and how it was trained, into a continuous mathematical space. So the number of dimensions vary depending on the model. Each model has a different number. We have protein language models that have like 300. We have some others that have like over 5,000. Most of them have around 1,000 or 2,000. And some important features of these embeddings is that they do have a magnitude that we call the norm, and they do have an angle that we call the direction, right? So just imagine this is in all these dimensions, we can still measure this. So the idea is that we can convert sequences into vectors, and then we can operate on the vectors to see how similar or dissimilar they are. So it was mentioned in Annabelle's talk this morning, but some of the most advanced ones use transformer architecture, which is basically AI model designed to pay attention, to understand the context. And one classical example to understand how it works in terms of language, human language, is the word eat. That could mean many things, right? So the cat sat on the mat because it was tired refers to the cat, but the cat sat on the mat because it was cozy refers to the mat. So if we pay attention to the context, we are able to understand what it means in each case. So this is what these models try to do also at the level of protein language models. So they look again at not only at each position at a time, but at all the context. There are tons of pre-trained models, many of them. This is, and they are divided in families, like big groups that have the same characteristics, let's say. So I'm going to put one example here. One of the first ones, I think, was developed in 2021 by the team of Burkhard Rost in Munich, and it's called the ProTrans, just to give you an idea of some of the features. So this was an adaptation of popular natural language protein models, such as BERT5 or BERT or T5. They are trained on very large-scale data sets. This in particular is trained on the Big Fantastic database, which is all UniRef and Magnify, which is microbes and soil, metagenomic data, and so on and so forth. So a huge amount of information. And there are different versions of it. Some don't have transformers, some others have transformers, and some use different tokens to encode the protein. I will tell you a little bit more about this in a minute. And something that is also very different sometimes is the architecture, which could be an encoder, a decoder, or an encoder, decoder. And again, I will tell you a little bit more about this in a minute. Other protein language model families that you have probably heard about is the ESM, the Evolutionary Scale Modeling, developed by Facebook, AI Research, ProGPT by the lab of Noelia Ferruz, UniRep by the George Church Lab, ANC also by the Rosa Lab, ProGene by Salesforce, which is another company, and so on and so forth. So in this table, I'm showing you some of these features that make the protein language model families different, right? And I think this is an important thing to keep in mind because they vary a lot. It's not the same learning Latin language that you just need 21 letters than learning Chinese, that to have a conversation you need to learn 3,000 characters, right? So think about these language models as something like that. So I'm just putting here some examples, ESM by Meta or FAIR like Facebook, AI Research, ProTrans, and ProGene. So the training objective is, or the unit, let's say, well, first of all, they are trained on different data sets. The ProTrans, it's trained on this giant, big, fantastic data set that I told you about. The ESM is trained on a smaller database. And something that also differs between them is the training unit. So for instance, the ProTrans takes into account chunks of amino acids in a continuous manner at the same time to do the encoding. And the ESM goes like position per position. ProGen does it in an autoregressive manner and so on. So this can vary a lot between language models. The architecture, as I was anticipating before, also impacts a lot with these three main architectures being key. We can have either an encoder, which is a reader. It's taking these chunks, these tokens, and then converting them into an embedding and focusing on this first translation rather than putting a lot of attention into it, which has been proven to be quite good if your interest is analysis and understanding what type of, the encoding of sequences into these vectors. Then we can have on the other side the decoder, which is, let's say, going back from the embedding to the sequence. And this is called the writer. And this is great, for instance, for generative AI, right? Like predicting the next best amino acid or sequence, like going backwards. And then we have something in between, like two layers of encoding that's called encoder-decoder. That's, let's say, the translator that explores both things. Like first, how well the encoding is from your sequence to the embedding, and then it circles back from the embedding to the sequence to double check that everything was fine. So this is a little fast introduction to protein language models. Again, I think the take-home message is that they can vary a lot in what they are trained, in their architecture, and in their internal way of working. So it's very difficult to generalize, right? So we need to be quite specific. I think it's important to keep in mind that the conclusions will be specific to that specific language model. So I'm going to tell you a little bit now about the internal work of this model, Protify, which is the one that we are mostly working in because it seems to work better for our purposes. And it all starts, of course, with a sequence. The first step is doing these chunks of amino acids together in a continued mode and encoding them. So after the encoding, what happens is that there are a series of layers of learning. In this case, in the Protein there are 24 layers. Each model has a different number of layers, like EVO2, for instance, the genome model has 32. Some others have 12, etc. So what happens here is that for each of these chunks that are encoded, in each of the layers, the model is learning the context, and it can be learning different things in different layers and in different chunks. It may be learning what the neighbors are, what the most frequent neighbors are, or it may learning that there's alpha helix 20 amino acids upstream, or it may be learning that it belongs to a Drosophila, right? Like we don't know yet exactly what's happening here. There's a lot of research, and there are ways of trying to figure it out. And then what happens is that each layer generates one embedding with whatever has been learned. So we have all the embeddings from the different layers. I'm bringing this up because there seems to be a relationship between the layer and the type of information that is learned, with some layers having more phylogenetic information and some others having more structural information, as we will see later. And then what happens is that we have a concatenation and then a mean pooling. So at the end, we feed our sequences here, and we get usually a mean pooling. There are some softwares that allow us to get the layers if we are interested. And then we have tons of vectors to work with, right? We do our classification task or whatever we want to do with them. So this is how it works. So what we did, again, we studied 2021, 2022, was, okay, let's leverage these language models and convert our protein sequences from invertebrates and non-model organisms that we don't know what they do into embeddings. And then let's try to plot them in the embedding space and to calculate the distance between the neighbors. And if we have a neighbor with a Go term in uniprot whatever, we can then assume that it has the same grammar and similar function, and we transfer the Go terms. So what we did is to develop this pipeline called Fantasia, functional annotation based on embedding space similarity. We benchmark it in a bunch of model organisms with different models. And then this is like getting cut all the time. I'm just going to do this the last time and reinitiate it because it's getting cut. There we go. And then, yeah, we do have the pipeline up and running, so you can feed a full proteome, like 30K, 40K genes, whatever you want, and it converts these into embeddings, and then it calculates the distance to sequences that have Go terms from reference databases, and then you can have the Go term, let's say, propagation. And at the end of the output, you have not only the embeddings of your sequences, but you also have putative Go terms based on these neighbors. So we have these plots in the paper where we explore what we call the dark proteome, right? Like genes that before did not have any functional annotation, and we wanted to see them for the first time, right? And just as let's say, explore their potential as hypothesis generators. So we took tardigrades as one iconic example of cute animals, but also extremely resistant to many conditions, as you know, desiccation and heat and so on and so forth. And what we did is go enrichment. We took the genes that were only annotated with Fantasia, but not with homology, so the ones that we did not know anything about. And we did an enrichment against all the genome. Many functions are common, related to the immune system or reproduction, development, and so on. But we also saw one category, that is detection of stimulus, that was involved in a response to UV light, water deprivation, heat, osmotic stress, DNA damage, and so on and so forth, which we just put as an example of genes that through the function you can use as hypothesis generator to test your, go on into the lab and do some experiments or check how these genes have evolved and so on and so forth, because they may be perhaps potentially involved in the extreme adaptation to hard conditions in these animals. So this is basically what I presented a couple of years ago. And what we realized is, okay, now we have like literally millions of embeddings. We are seeing that they do have a lot of signal. We are seeing that you can generate them super fast, because I did not mention that, but this pipeline works at, let's say, 20 proteins per second. Like you can encode in a super fast manner hundreds of genomes. Like we did 800 genomes overnight, which was like 20 million genes. Like we have now millions and millions of these. Like the important thing now and the most challenging is to curate your structural annotation, right, before you feed it here. But then it's super fast. So we started to think, okay, can we perhaps use these embeddings to tackle some problems in phylogenetics or comparative genomics? So in other words, let's test whether they have phylogenetic signal, because that would be very cool, right? Particularly now, we need to scale up our methods with all this amount of information that we have. So what we did is, again, go back one step. We have all code terms, but we do also have the embeddings. Okay, so let's see what we can do with them. We were using the Proti5 with 1,024 dimensions. So we started to look in the literature what people were doing in terms of using protein language models and phylogenetics, and we did not find a lot of information. So one of the first papers was published in 2022. It's this paper saying that protein language models trained on multiple sequence alignments learn phylogenetic relationships. So these authors were using MSA Transformer, which is a language model that was developed in 2021 by the Facebook people as well, and some people at Stanford. And basically you input multiple sequence alignment. There were some questions before about multiple sequence alignments and language models. You can indeed use attention layers in multiple sequence alignments and get an embedding out of it. And they were doing that, and then they were building embedding trees with these embeddings and comparing them with the having distance in the multiple sequence alignments, and they were seeing that there was good signal. And they were showing, there's a paper in Nature Ecology and Evolution, Nature Communication, something like that. They were also showing that these models can separate coevolutionary signals and separate function from structural constraints. So that was, I think, one of the first ones dealing with this. Then, very recently, like in January 2025, there was this paper by the team of Michael Boden, who is doing a lot of work on using protein language embedding, well, testing whether protein language embeddings encode phylogenetic signal. And what they were doing is to test different protein language models. Remember, I told you they are different in how they are trained, they are different in how they work in terms of the architecture, in their learning units, right? So we should expect different results. And what they were getting is that indeed they were having phylogenetic signal mostly in early layers. One of the novelties that they introduced is the analysis of individual layers. Remember, I was telling you they learn in different layers. So they were doing analysis with different chunks of layers, and what they found is that the first layers apparently were dedicated to learning sequence similarity / phylogenetic relationships, and the later layers were mostly involved in structure. And they were finding that they were particularly good for deep divergences, but that they were sensitive to gaps. All these papers were based on individual gene trees for gene families, for PFAM families, with or without multiple sequence alignment. Then, January this year, there was this other paper from the same team, focused on more thorough investigation on the MSA transformer and getting embeddings from alignments, also in individual gene families. And they were getting pretty much similar results, but with, let's say, more thorough investigation. And basically, they were saying that they use epistasis across sites and gaps that were ignored by classical methods. So they are opening the door to these methods being able to perhaps overcome some of the limitations of classical molecular evolution methods, and that they can even recover novel evolutionary relationships at the level of gene family. Okay, so there are a few more papers out there, but the truth is that there are not so many still of people using embeddings to test whether one can recover phylogenetic question. So I personally think this is really exciting. And this could perhaps be we may be witnessing a new era, a new beginning to investigate in a systematic manner this embedding analysis through the lens of evolution. And this is truly exciting. So we wanted to contribute to these debates with a species tree reconstruction, which is a topic that at least I have not seen yet in any publication. So in our case, what we decided is to not use alignments and went straight to the embeddings, as I will tell you in a minute. So this is a project that has been led by Alexander and Belen in my lab, with the collaboration of Alex and Ana Rojas in Ana's lab, mostly because now we have implemented in Fantasia the option to extract specific layers, embedding from specific layers. So I think this is going to be very cool. So what we did is quite simple. Just as a disclaimer, in my lab, we are not expert on method development. We are, I would like to define us mostly as empirical testers, but we are happy to go kind of midway when there are not methods that fit our needs, right? So you may not see the most sophisticated methods here. They are conceptually pretty simple, and that's actually the reason why I wanted to present these ideas to you, because I think that this can be a great opportunity to get feedback on how perhaps to improve this work. So what we did is to take two species trees, one that we call deep divergence. So we have animal phyla level, 8 tips. We have an outgroup, unicellular outgroup of animals, and then we have sponges, cnidarians, craniates, mollusks, annelids, arthropods, and priapulans, one of each. And then we have what we call the shallow divergence data set, that is reptiles with the outgroup, the platypus. We have like salamanders and turtles, tortoise, and then alligators. So we did 3 approaches. The first one is the multi-species coalescent one. So what we did is, okay, let's start by orthogroups. We infer orthogroups from these ones, but what we did is to replace the sequences by the embedding. So after this, we went ahead and built a distance matrix between species. And let me remind you about the embedding structure. Remember, we have a norm, that is the magnitude, and then we have the angle, that it's the distance, a way of measuring the distance, positional distance, let's say. So at that point, we're a little bit puzzled how to continue, like what was the best distance to take into account, particularly whether to normalize by the distance or not, because it's not super clear what the norm, the magnitude, means. So it's not directly interpretable, and it emerges from the model, and it can reflect different factors. It can reflect sequence properties, but it can also reflect noise. So apparently, the higher magnitude, the higher noise, but it could also mean the higher distance based on a few papers. The problem is that, again, it's not comparable because they are different protein language models and different layers. So what we do know nowadays is it may capture both biological signal and technical bias. So since we did not know, what we did is to analyze two types of data sets, one with one normalized, where we are only taking into account the angle between embeddings, the direction, and the other non-normalized, where we are taking into account the direction and the magnitude. And we went ahead, and with this distance matrix, we built embedding trees. So, of course, at this point, if we were working with sequences, before showing you the tomato, let me tell you why I want to stop here a minute, because, of course, if we were working with sequences, we will think on substitution models, right? But the thing is that protein language models are supposed to be trained in huge data sets. So the idea, or the hypothesis, better said, is that you may not need a substitution model because it may have been learned implicitly. So perhaps just by analyzing the distance, we are already implicitly integrating all this information. And I put here a tomato because when I told people in my lab the type of conference and the type of audience, they made a bet that people were going to throw tomatoes at me after this slide. If you don't do it, that's okay, because there will be a second slide where you will have the chance of throwing a tomato. Okay, so again, this is just a hypothesis. We are just testing how things work. So we built individual embedding trees, astral, and compare Robinson fold distances. We did different data sets from single copy. This is the taxon occupancy. So genes present in all the species and genes present in one of the species. So from, let's say, 18K to 700, more or less. Of course, we have paralogs. What did we do? First, we analyzed everything with paralogs, and then we decided to choose one sequence close per species, close to the centroid of the ortho group, close to the species centroid, or building a synthetic embedding that was like the real centroid. We did this for different layers and for them in pooling. Again, remember that there was this evidence saying that early layers encode phylogeny, late layers structure, and we did the normalized, non-normalized. Second approach, supermatrix, orthogroups, embeddings, again, concatenation, distant matrix, where we build embedding trees and compare Robinson fold distance. Very similar to the previous one, but just concatenating. We just did two data sets, the single copy and the orthogroups in all species, even with paralogs, because, disclaimer, we did not see a lot of difference in analyzing 2,000 present in all the species or 18,000. And we did the same measurements, species centroid, synthetic embedding, closest to the orthogroup centroid, closest to the species, layers, and normalized. And then the third approach, the most risky one, perhaps, a more exploratory one, and where you can throw a second tomato if you want, is the cloud approach. So what we did is to forget about orthogroups, forget about alignments, and we thought, what if we just encode each species as a cloud of embeddings? So we took this, we took all longest isoforms from each species. So just imagine how many genes does a sponge have? 30,000. Then you have the longest isoform. For each gene, you have 30,000 embeddings. And you compare it with, I don't know, 20,000 of an Iderian, and so on and so forth. We measure distances. Here, we explore 4 different distances. Energy distance, that is the most straightforward, inter versus intra. Maximum mean discrepancy, that uses a kernel. I'm running a little bit late, so I'm just not going to stop here. Was-sustained distance, that it's like moving each point in one distribution to the other one and measuring the cost. And the Fréchet inception distance, which is a distance developed for generative adversarial networks, mostly to compare images and networks and so on. And we thought that could be mimicking our cloud distribution. Then we build trees, and then we compare the Robinson fold distance with the species tree. And here we did all the embeddings, a subset of 3,000 per species, all the layers, and the normalization. So, results. Regarding the multi-species coalescence, in the deep divergence, we have this type of graphs called radar graphs, where in this axis we have the Robinson fold distance, so closer to 0 the better. And in each of the axes we have the different data set, 18,000K, 11K, 9.4, depending on the taxon occupancy, and here we have the layers and the single copy orthologs. So what we found is that it was pretty bad. Robinson fold distance was 4. Always the same 2 or 3 species were jumping. Single copy genes was worse than the multi-copy genes. No difference between normalized and non-normalized. And early mid layers were slightly better. And the best results were in terms of removing paralogs regardless of the method. So choosing one species close to the centroid, one sequence close to the centroid worked better than non-removing paralogs. The shallow divergence data set worked a little bit better, but still with a Robinson fold distance of 2, we did not get the perfect tree. Single copy orthologs, in this case, they were a little bit better. Again, these animals are much more similar to each other. No difference between normalized and non-normalized. So the magnitude here was not particularly important, and no difference between layers. So actually the layers were worse than the mean pooling. And the best results were, again, closest to species centroid, non-normalized. So choosing one species, one sequence for your paralogs. Then, supermatrix approach, regarding the deep divergence data sets, we got our first bingo. So we got that the layer number 3, when we were removing paralogs and just choosing the sequence that was closest to the species centroid, was giving us the exact species tree. Reminding you, these are different animal phyla. So we still don't have the results with sequences, but I can put my hand in the fire based on my experience in phylogenomics in animals that we are not going to get this with sequences. Early layers working better, layer number 3. Single copy orthologs are pretty bad. And normalization in this case had a bad effect because the normalized data set gave Robinson fold distance of 4. So the magnitude of the vectors were important. In the shallow distance divergence data set, we did not, we got kind of bad results with a Robinson fold mean distance of 4. The single copy orthologs were equally good or equally bad than the other data sets. No difference between normalized and non-normalized. And the layers worked even worse than the mean pooling in this case, probably because the sequences are more similar, because we are dealing with more similar species, and what the embeddings are encoding, it's perhaps differences in structure or something like that. So the best results were with single copy and all OGs, but again, not perfect. Now, regarding the embedding cloud approach, remember, no orthogroups, no multiple sequence alignments here. With the deep divergence data sets, we got like, well, first of all, we were also evaluating how the different measurements of distance were working. We found a huge difference. We did not get the perfect Robinson fold distance of 0 to the species tree, but not too bad. We got like 2. With the Wasserstein distance, layer 3, again, normalized, and the Fréchette layers 3 and 9 normalized. So here normalization matters, again, early layers having more signal. Best distance was the Fréchette. Worst distance, pretty bad when you see a lot of color, is that we have a lot of Robinson fold distance. Normalization had a big effect because here it was better not to normalize. So the normalization ones, like here, there's a huge Robinson fold distance. So let me go a little bit deeper into this other representation that's perhaps more clear. So here we have the different distance metrics for the subset of 3,000 random embeddings and for all the data sets. So this is energy-based, like a lot of color, but again, for some early layers, either normalized or non-normalized work better. The Wasser stain, this is 3,000 genes, this is all of them. There's a lot of whites, particularly in early layers, regardless of normalization, meaning that it seems to work quite fine. The Fréchette, here we see the other way round. Early layers, non-normalization, huge Robinson false distance, and less in the normalized ones, and the MMD pretty bad. And if we plot the layers at the end with all the different measurements, what we see is, again, layer 3 normalized and layer 9, like early layers, show lower Robinson fold distance towards the species tree compared to the other ones. And the later ones are pretty bad, like 21. Regarding the shallow data sets, we got our second bingo because with the Wasser stain distance in layer 3, non-normalized this time, we got a Robinson false distance of 0. The best distance seemed to be the Wasser stain with all the embeddings, again, non-normalized. So here the magnitude is more important. Again, there's some more similar sets of sequences. The rest of the sequences are pretty bad. You see a lot of color. And the normalization seemed to have a big effect in some of the distances. So if we zoom in into the measurements, again, measurement methods, energy distance, pretty bad, a lot of color. Fréchette, a lot of color as well. MMD, kind of depending on the layer, and the Wasser stain here was kind of the best for all data sets, particularly for layer 3. And regarding, if we pay attention to the layers, it kind of varied. So in this case, the layer was less important. The method of measuring distances was more important, with some chunks that are white, and meaning that the method work, the measurement distance method performed better, and some chunks having a lot of color, meaning that it was not recovering. Well, the Robinson false distance was very high. And I think this is pretty much it regarding the take-home messages. Protein language models, at least this one, Proti5, because again, they are all very, very different, and we should not be comparing them. It seems to recover phylogenetic signal with embedding trees and embedding cloud trees sometimes being fully congruent with the species tree without the need of multiple sequence alignments or even orthogroups. But of course, the results are varying a lot depending on the approach and the data set. Regarding the approach of the multi-species coalescence, where we build orthogroups and then we replace them by their embeddings, and we build embedding trees, it seems to work relatively fine for shallow divergences, but not for deep ones in our n=2 trees that we tried. Regarding the supermatrix approach, what we found is that it seems to work well for the divergences, particularly for early layers. And removing paralogs via the selection of the closest embedding to the species centroid seems to yield the optimal results. The embeddings could approach cloud approach, so no orthogroups, no multiple sequence alignment, work quite well for both data sets, but with different layers and different normalization steps, with the Wasser stain and Fréchette performing better than the other ones. So this is again a very first empirical testing on the performance of one of these language models to try to infer species trees. There are still more questions than answers, but I think it's, I don't know how you feel about that. I feel very exciting times coming. And I just wanted to finish by bringing to the table the debate of whether we should be perhaps thinking on how we can use protein language model embeddings to create perhaps hybrid systems with the classical molecular evolution approaches to bypass some of the traditional bottlenecks that we are finding. And this is it on my side. I would like to thank all the cool people from the lab. I'm also in front of the sea, so I'm not jealous of your views. and all our sources of funding. Thank you.